Does that mean retry?
Yeah, that might help.
Why do I have nothing on pathology?
All right, so yeah, I just went live. I don't know whoever's going to be watching this right now. The Coccidian quiz was due today. We're going to move that to Sunday night because we didn't quite finish. We didn't finish the lecture yet. So we'll move it to Sundays. That way you can take some time over the weekend. That is submitted. Let me check out.
Just a retry. Those of us that were a little overly ambitious.
I don't know how many people did it. Quite a few of you did it. Yeah. So, who's here? Amber, do you want to retry?
Yes, please.
Clear that up. Clear attendance. All right, Emily.
I didn't really get good in study.
Yes.
I made a good grade, but it wasn't really helpful in studying.
Let's see here. Olga's not here. Alexia. You want to retry?
Yeah.
What about Ashley?
I have a 23 too. I rolled
99%.
I thought we were going to finish Toxoplasma. Ashley. Hope.
Yes, please.
10. Skylar's not here either. Let's go to live stream. Here we are. Yep. All right, so live stream's up. There's just 2 other people. I'll just email them. Send an email right now, so I don't forget. All right. Well, ask them if they would like it to be clear. All right. Email sent off, so other 2 people that didn't, they took it. They have an opportunity to email me. All right, so Toxoplasma gondii, this is where we left off. We finished up the life cycle, right? We've got this enteroepithelial life cycle that's in our cat. Right, and then we have this extraintestinal cycle, which is in our cat and other hosts. Once we get into this extraintestinal cycle, once you're infected, you're basically infected for life because those bradyzoites are going to be hiding inside of a zoedocyst. And that wall that gets laid down is going to be relatively impervious to the host's immune system. So, when we think about life cycle adaptations or adaptations to evade the host immune system, being intracellular is one. But you do have to kind of figure out how you're going to prevent the cell from tagging itself to say, hey, something's wrong with me, clear me out. All right? So being inside works. Pathology. All right, so most of our infections, we didn't talk about this, did we?
Just a little bit.
A little bit, yeah.
Did we?
We started going over it. I just don't have what's left.
Okay. Yeah, so most of the infections in humans, you're going to be mild or asymptomatic, and they're going to pick up early on, unless some sort of stressful event happens and all of a sudden you'll have a couple of zoedocysts that rupture, releasing bradyzoites, and then your memory response takes over, ramps up, clears out the bradyzoites that are circulating. Hopefully, you'll clear out most of them, but some of them will reinfect. In the acute infection, it's basically symptoms of flu. And this has affected people. I mean, you can imagine that it's affected people, and some people, very prominent people. Who knows who Martina Navratilova is? Anybody? Sports buffs? Martina Navratilova, tennis star. Broke in the late 70s, dominated throughout the 80s. She's now some human rights causes, but look at this. What was it? She holds the best season win-loss record in the Open era, 86-1 in 1983, and 4 out of the top 6 Open eras. She recorded the longest winning streak in the Open era. Over 5 consecutive seasons, she won 428 out of 442 singles matches. One of those was in the US Open in 1982. She lost in the quarterfinals. Why did she lose in the quarterfinals? Which one? She suffered from an acute Toxoplasma infection. Really knocked her out, but she still went out there and competed. So it was pretty impressive. It's going to be hard. It's going to be buried in the information that's what happened. But if you kind of look through here, all of these wins, all the opens. She was on a streak. Don't have the U.S. Open on there. So this was championships. Here's all of her. She went to the quarterfinals in 1982. She should have. She was healthy. She probably would have won it. So, yeah, parasites are non-discriminate. You're gonna hit anybody. So those are acute infections. Chronic infections, if you have it, and if they continually rupture, you will get some localized necrosis happening, and depending on where those oedesis form, you have these various types of symptoms, some major symptoms. All right, did you get these? I think we did cover this. All right, congenital toxoplasmosis is the major concern.
This is one of the
Yep.
What's that?
This is one of the questions on the quiz.
What is that?
All right, so it results from vertical transmission. So we talked about horizontal and vertical transmission. Horizontal is me giving it to somebody else. Vertical transmission is when a parent passes it to their offspring, or a grandparent. You can probably say grandparent passes it to their offspring, but in all the cases that we talk about, it's parent to offspring. All right, for Toxoplasma, we are talking about a mother acquiring infection and then passing it on to the developing fetus. All right, so it's not after the child is born. It is the baby is developing. All right. If the mother is pregnant and acquires Toxoplasma gondii, then about 45% of the time, the fetus will acquire it. All right. How does they get it? How do they get it? It's the tachyzoids and the bradyzoids. All right. Those are circulating. They'll get to the blood. Mother shares the blood with the fetus. All right. Now, of these 45% of cases, of getting it, 60% will be subclinical. You probably wouldn't know that the fetus has it. Right? Probably wouldn't know unless the fetus comes out and you took a blood sample. All right, you have to wait for them to develop. Say wait a year or so if they're not breastfeeding, because we don't want any maternal antibodies in there, and then maybe you can detect antibodies against Toxoplasma. All right, 9% of them often result in spontaneous abortion. So you think about individuals that you may know that lost a child early on in pregnancy. Some of that could be possibly due to Toxoplasma gondii. we don't know, kind of have to go out and do a lot of stuff. And quite frankly, it's probably such a traumatic event that you probably aren't going to have doctors and researchers probing the parents at that point. The other 30%, and I know we don't add up to 100, rounding errors, 30% develop severe disease. All right, types of diseases that we're talking about are hydrocephalus, intracerebral calcification, severe mental retardation. Well, a range of mental retardation. All right, hydrocephalus is fluid around the brain, results in a large head and normally a small brain. It impedes development. As part of this, you can have calcification reactions happening. So you'd get maybe a zooidocyst in there cause, ruptures, the immune system comes in, starts calcifying it. And again, now you're destroying portions of the brain where the brain should be developing. If the child is born, then death could happen, shortly after birth, or could happen several years down the road. And in a lot of these cases that have these severe disease, if the child is born and survives, they typically exhibit the mental retardation. All right, this is kind of a major concern. This is why you hear that pregnant women should not clean litter boxes. But that's only part of the story, because consumption of oocysts, or acquisition of the parasite by accidental consumption of oocysts, is probably a small part of the total infections. Chances you get it is going to be pretty small, because typically you clean the litter box, you're probably going to be washing your hands before you start preparing food or cooking or anything. And when you think about how long cats shed the oocyst, they're not really shedding for very long. More likely scenarios are you're out gardening without gloves, you pick up oocysts that were deposited by wild cats. And now, without even thinking, you grab that tomato off the vine and you eat it. Looks tempting when it has the dew on it. You think, oh yeah, the water's clean. You don't know that. Water could have hit the ground, splashed an oocyst on it, and you can get it. So gardening without gloves, eating raw meat or undercooked meat is probably the most common way in which you get it. So pregnant women, if you're going to have raw meat, know that this is a risk, and it's gonna be red meat. You could, some cows carry it, but we're talking beef. Cook it to kill the zooidocysts to avoid that infection.
You're saying don't eat rare steak?
Probably no. They do. So we don't have any ag students here, but in animal production, they do test for Toxoplasma. I believe it's an ELISA test for serum. But they don't test. I don't think they test every animal. They test a lot, small sample, as a representative of the entire thing, representative of the entire lot. So our food supplies, okay. But this is part of the reason, like beef tartare is at least attributing to increases in Toxoplasma prevalence in countries where that has become popular among the younger generation. I guess your generation and a little bit older. All right, so wash your hands if you're out in the garden, cook food. Other information. All right. Once the zooidocysts start forming, we're moving into the chronic infection. The infection is going to be persistent. And those zooidocysts will occasionally rupture, releasing additional bradyzoites. All right. When these start to appear is typically when you start seeing protective immunity develop. Right? The host immune system starts to learn what it needs to fight. One of the interesting things about this is that we develop premonition. These bradyzoites that come out of the zooidocysts serve to protect you from getting a super infection. All right, so you get it, you exhibit this, these flu-like symptoms for a week or so, maybe 2 weeks, and then your body clears it out, and these bradyzoites and the zooidocysts help to keep you from having it again. All right, so premonition, we've seen it, we're going to see it again. All right, it's just repeated exposure. Our body is successful at clearing out the parasites, but some of those bradyzoites escape, get inside a cell, and then can hang out until they rupture the cell again. That's different from concomitant immunity. As a review, what's concomitant immunity?
Is that when you have a disease and it prevents you from getting other diseases?
Yeah, it's just like premonition, right? Prevents our super infection, and it can prevent us from getting some other stuff. But how is that different from? How is concomitant immunity different from premonition?
Is it like you get infected multiple times by the same thing? Like you're not
Premonition, you could.
Yeah. And so is that the difference with the other one? You're like you actively have it, and that's what's keeping you from getting it?
Yeah, possibly. What parasite exhibited concomitant immunity? One of the early ones. Take a guess. Carter, take a guess. Take a guess. Well, just name a parasite that we've covered and did a life cycle on. No, good guess. That's not it. Kayla, take a guess. Ascaris is off our list.
Okay. The hookworm.
Nope, hookworms are off. I'm going to say we need to go earlier than that. Who's going to guess next? Alexia, take a guess. Name a parasite. Before our hookworms and ascaris.
I can't think of any because of how many we cover.
How many we covered?
It is a nematode, right?
No. It's not a nematode. So then what's the other group that we talked about before the nematodes? The what?
Flatworm.
Flatworms. All right. Schistosoma. Concomitant immunity. Are adults in there? The adults are also confer protection, confer apparent immunity against a super infection. Difference though is that the host immune system doesn't even touch schistosoma. Parasite's unharmed. Whereas here in premonition, yeah, the parasite's harmed. We recognize it, we clear it out. It's just that the problem is that, some of those bradyzoites escape by reinfecting cells. All right, so we do see this premonition, we do see protection against super infection, so that we'll see premonition again with Plasmodium. The other thing is that the parasite seems to alter the behavior of these intermediate hosts. All right, so we talk about, if we're talking about adaptations to increase transmission, this could be a behavioral change. And the behavioral change that we see is that infected individuals appear to be reckless in their behaviors. All right. In terms of ecology or whatever, some would call it fearless. All right. So infected mice, for example. Right? If you take a regular mouse and you let them run around, they're going to be hesitant when they come out into the open. They're very hesitant. They're scanning the area. Infected mice do not do that. Some mice, some rats, right? They smell cat urine. They avoid it. They're fearful of it. Infected mice, not so much. Infected rats, not so much. All right, how does that happen? Well, it seems like the parasite infects the dendritic cells and causes them to produce and secrete GABA. What's GABA? Look it up. You've seen it before, right?
Yeah.
Oh, I have it written down.
show us all of the aquatic vertebrae? Or like the fish?
We didn't call it in here. We didn't call it in here. It would have been someplace, so I'd have to, let's look it up.
Gamma aminobutyric acid.
Yep. All right, what's its function? Where would you have encountered it before? Okay, it's a neurotransmitter. All right, so if you had physiology, you probably talk about it. Human phys probably talks about it. So yeah, it basically secreting this neurotransmitter, and that neurotransmitter is going to reduce fear and anxiety. At least that's kind of what we know, in terms of humans. And I think you can also replicate that in mice. So the parasite seems to be adjusting these. Now, what are the dendritic cells doing? They're probably picking up some of these parasites. The dendritic cells can be phagocytic to help clear it out. But we know that this is probably what's happening. And that means then that in humans, we probably exhibit something similar. The hypothesis out there is that some of these reckless people may actually be suffering from lifelong toxoplasmosis.
How do you tell someone who acts reckless, like, you know what? I can be.
Be bold, tell them that. And what can you do about it? I mean, I don't know. It's a hypothesis. Same thing with there's also a hypothesis that people that are schizophrenic might also have toxoplasma, that it affected their brain in some way, and it's just kind of based on this idea that I know Carter is normal now, but in a year from now he could be schizophrenic. It's like all of a sudden it just happens. Like what causes that? Maybe it's a parasite. Maybe it's some other infectious agent that caused that to happen. Who knows? All right, questions? That wraps up our coccidians. So now, let's get to our final presentation. And we will have to finish this on Monday. That way, we can go live for our exam. So practical goes live on Monday. You'll have all week in lab. We'll use that time. You can go in and review the slides. Review any of the slides that we have. I'll have the 3 quizzes that we had out along with keys. So if you want to go back and check those. So this is our last part of our protozoans. All right. So now we're in the class Aconoidasida. So we were in the Aconoidasida and the Eucoccidians for Toxoplasma. Now we're in the Aconoidasida. So generally, the conoids are going to be absent. Hence the name, Aconoidasida. The other thing is that syzygy is absent. We're not going to have these gametes that fuse. Instead, we'll see micro and macro gametocytes that develop independently. You're going to have gametogeny happening to produce micro and macro gametocytes that will ultimately produce your gametes. All right, the microgametocyte is going to produce flagellated gametes that will then fertilize the macrogamete. So one microgametocyte is going to produce multiple microgametes. One macrogametocyte remains one macrogametocyte until it gets fertilized, and then it becomes a zygote. Now, the zygotes in this class are motile, so they take on a different name called an ookinete. The ookinete will deposit some sort of cyst. It's not going to be like a hard cyst wall like our coccidians. Right? But it's a little bit thicker than just a plasma membrane. And inside of that, so the ookinete produces, that would be our equivalent to our oocysts. All right, once it gets that membrane down, and then you will have the sporulation and sporogony to produce the sporozoites. Now, our sporozoites will not be inside of a sporocyst. So in our Toxoplasma eimmaria, you had the oocyst that underwent sporulation to produce your spore cysts, and then sporogony to produce the sporozoites inside of those spore cysts. Not so in this class. We skip that spore cyst stage. All right. The order that we're going to talk about is the order of Hemospororida. Hemospororida. This blood sporidium. These have a heteroxynous life cycle. For us, merozoites are going to be produced in the vertebrate host. The sporozoites are going to be produced inside of an invertebrate host. Oftentimes, it's going to be an insect, but it doesn't have to be. It could be something like a leech in some of these Hemosporidiums. And most of the parasites are in wild animals, have little effect on humans. We get careless. A lot of people would care less. But we do have one of these that is pretty significant parasite of humans. Oh, we got two. We had Babesia that was presented. All right, ready? All right. So inside the family, Plasmodiidae is the malaria and malaria-like organism. The genus that we're going to talk about is Plasmodium. This is the causative agent of malaria. Malaria got its name for bad air. It was termed that because ancient Romans, associated these types of symptoms that we would get with wetlands, marshland environments. In wetland, marshland environments, some of these areas, you have a lot of methane being produced in the substrate area getting released. It takes on a noticeable smell that they would classify as bad air. Right? So that's how they called it malaria. If you think about it, those areas are associated with mosquitoes who are going to be spreading malaria. They're the insect vector. All right. So human infections are a major problem. Most up-to-date numbers comes from the CDC. So in 2019, we had 229 million clinical cases of malaria. 409,000 individuals died of malaria. Most of these deaths are in children in Africa. It's a huge burden on Africans. About 2,000 of the cases are diagnosed in the U.S. All right, and these are from either U.S. travelers or immigrants that migrated over here. We don't think we have had a confirmed transmission event here in the United States in decades. We'll mention that near the end. Ready? All right, life cycle. Humans are the vertebrate host. Mosquitoes are going to be the definitive host. And as I have on our slide here, the life cycle is going to be broken up into two main parts. But we can get those parts all on the same graph. So we are going to do the life cycle of Plasmodium species. We have 4 Plasmodium species that infect humans or can cause pathology in humans. They all have a similar life cycle. So for Plasmodium species, we're going to start where we have our sporozoites. They're going to be in the salivary gland of our mosquito. And the mosquito is going to feed on the human and inject the sporozoites into the blood and tissue. Blood and tissue. These sporozoites will then utilize the blood and migrate to the liver. Migrate to the liver. Once we get to the liver, then these sporozoites will penetrate into hepatocytes. Hepatocytes are liver cells. I could have written liver cells, but hepatocytes. Increase your vocab. All right. So, inside our liver cell, did this on purpose, right? We will transform into a trophozoite. Right, so we are in a liver cell, all right, and then in our trophozoite, it'll feed, it'll develop, and then when it gets ready for our transition, when we get ready to transform or undergo merogeny, it transitions into something called a cryptozoite. I didn't add it on here because I'm not going to ask that part of it. The cryptozoite is basically the process of producing all of the apical complexes, all of the extra mitochondria, everything that's going to go into the merozoites, because then once everything gets prepared, it undergoes the merogeny to produce our merozoites. All right, so the cryptozoite is just a fancy term for a certain stage that's undergoing process, the getting ready to replicate. And this is merogeny is again our multiple fission event. All right, so we have merozoites in the liver cell, and then that is going to rupture and release the merozoites into the blood. All right, so from the blood then, we will now, these merozoites will recognize and penetrate red blood cells. Right? RBCs, penetrate red blood cells, where inside our red blood cell we transform into a trophozoite. It'll undergo merogeny. To produce our merozoites that will then cause the cell to rupture and reinfect other red blood cells. Right? So, two different things right now. First thing, sporozoites went to the liver and infected hepatocytes. That part of our life cycle is called the pre-erythrocytic life cycle. Or pre-erythrocytic cycle. Right? Because red blood cells are erythrocytes. Or it has another name, which is primary exoerythrocytic schizogony. And here's where it kind of gets a little bit screwy, because I use the schizogony here, all right, but I use merogeny here. Merogeny produces the merozoites, but when you look up life cycle diagrams and read the life cycles in the book, oftentimes they talk about schizogony happening, the developing schizon. So you've got your trophozoite transitions to a schizon to undergo schizogony to produce our merozoites, and it's like, come on, man, just get the terminology right. So, we're going to use merogeny, merozoites, and so forth. All right. So this is the pre-erythrocytic life cycle, which means once we get into our red blood cells, now we are in the erythrocytic cycle. All right. The EE cycle is how you might see it. Good. all right? So we're in these two things. This rupturing is going to be an important source of pathology, and it is circular. It's going to keep going until either we get treated or the host will die. Now, some of our merozoites that get released, and it's going to take a couple generations, but then some of our merozoites will rupture and reinfect other red blood cells. And in those red blood cells, then, they will undergo gametogeny. All right, they will undergo gametogeny to produce a macrogametocyte, or one of them will produce a macrogametocyte, and the other one will produce a microgametocyte. All right, these are also termed macrogametes and microgametes. Again, it just kind of depends on which life cycle website you are looking at. Two different names. So microgametocyte, macrogametocyte. These are the stages that will appear in our peripheral blood. And these are the stages that will get consumed when a mosquito takes a blood meal. There's our mosquito, here's our human. All right, so now once we have that blood meal, we have our macrogametocyte in the gut. We have our microgametocyte in the gut. But what's going to happen is the microgametocyte will undergo multiple fission to produce our microgametes. All right? Microgametes. Now, what's the signal? How does a parasite know when to undergo this multiple fission? It's actually tied to pH. All right, so the red blood cell gets consumed. Multiple fission,
Yeah, sorry.
microgametes. All right, these are what they're called exflagellated microgametes because you have this microgametocyte which will all of a sudden kind of undergo this rosette-like structure under multiple fission. So the outer cell membrane, the outer plasma membrane starts pitching in, producing these 8 microgametes that are flagellated. All right. The trigger is increasing pH. So as the mosquito consumes red blood cells are going to be lysed in the gut, releasing the CO2 into the gut, which will bind to protons in that gut, raising the pH. That increase in pH is the signal that these microgametocytes need to undergo the exflagellation, that multiple fission event. All right, so that's how we keep it from occurring just in the body, in the human. All right, so now once we have these microgametes, these will find and penetrate a macrogametocyte to join up basically as a fertilization event. And that would produce our mobile zygote, the ookinete. That ookinete is in the gut, all right? And what it's going to do is penetrate and migrate to the hemocoel side of the gut. So it penetrates the gut epithelium to get to the hemocoel side. and the ookinete will actually remain attached to that hemocoel side of the gut. Once we get there, then it will secrete a capsule. So we have our ookinete on the on our hemocoel side, and then it will secrete what I'll put in quotation marks as the capsule. Again, it's not an oocyst wall, all right, and it's thicker than a regular cell membrane. But that capsule will basically cause it to produce our oocyst, which is still attached on the hemocoel side of the gut. Once we produce that capsule around it, then we can undergo our sporulation and sporogamy to produce our oocyst with sporozoites. All right, still on the hemocoel side of the gut, this will rupture to release our sporozoites that are now free in the hemocoel. All right, and once we get into the hemocoel, these sporozoites will migrate to the salivary gland. All right, once we get to the salivary gland, now we can be injected into our next host. Inside of our mosquito, once a mosquito picks up, so a mosquito has to pick up at least one of each. All right? It has to pick up one of each in order to produce our ookinete. But once we get them, once they pick it up, and typically they pick them up completely, then you're going to have these sporozoites appearing in the salivary gland in about 10 to 14 days. That's going to depend on temperature of the environment, of course. Cooler temperatures, it'll take longer to develop. It also depends on the species, and species of Plasmodium, species of mosquito that it's going into. But it's about 2 weeks or so. Mosquitoes infected. All right. Infected mosquitoes tend to feed more. So that seems to be a possible adaptation for transmission. It feeds more than uninfected. Now, is that a consequence of parasite siphoning off nutrients from the mosquito, so the mosquito needs to continue feeding? Maybe. Maybe it's some sort of behavioral change. Also, no, these are female mosquitoes. Why?
Aren't female mosquitoes the only ones that can
Female mosquitoes are the only ones that are going to be feeding. Why do they feed? Why do they need blood? They need the proteins for egg production. Males get by with just feeding on nectar. All right? These are female mosquitoes. One other aspect of this life cycle. So in Plasmodium, we can have relapse that occurs. All right, and I'm going to put relapse in quotation marks. Relapse. All right, because what I'm talking about is we have an apparent clearing of the infection, we're healthy, we're not exhibiting symptoms, and then all of a sudden, weeks, months, maybe years later, all of a sudden, we start exhibiting the symptoms of malaria again. All right, what causes that? Actually, 2 different processes. There is one that's called recrudescence that we'll talk about. Recrudescence happens when for some reason there's these merozoites or a trophozoite that's hiding out in a red blood cell, and all of a sudden it starts undergoing mirogamy and starts this cycle again. It's not a true type of relapse. They've called that term recrudescence because you're basically reactivating something that's in the red blood cell. At least that's the thought. True relapse is due to activation of another life cycle stage. So some of the sporozoites that get to the liver and penetrate hepatocytes will transform into a stage called hypnozoites. These are dormant stages inside of liver cells. They're just going to hang out there. They're going to hang out there. And at some point, we don't know why. We don't know what the trigger is. All of a sudden, these will transition, reactivate, and become a trophozoite and initiate the rest of the life cycle. This part is our true relapse. Our treatments, they don't kill off the hypnozoites. Treatments are targeting these merozoites in the red blood cell stage. So, that's why they ask, if you go to donate blood, have you had malaria before? And certain areas will exclude you if you've had malaria. We know the species, 2 species that infect humans, they'll produce these hypnozoites and we can exhibit true relapse. The other 2 species, they don't. You basically, after about 18 months or so, as long as you haven't had symptoms or you're not actively having symptoms, you could donate. Other species, the other 2 species exhibit this recrudescence. This presents a problem because we don't know which red blood cells have merozoites inside of those that are secreted. So this part, those species, those are definitely excluded.
Is there a time frame for that, or just you've never had it forever?
It should be forever. All right, questions? Leave that up there. All right, so almost. I think I mostly got everything. Let's tilt this down just for the bottom. Increased pH due to CO2 release from the red blood cell. That's our bottom part. All right. So I did send out an announcement, extra credit. Donate blood platelets plasma. When you do, you'll get a little sheet thanking you for donating, do's and don'ts, after your donation. Snap a picture, email it to me, I'll add 5%. All right, if you don't want to do it, or you can't donate, I do have this little story, or if you prefer to change it, I have a story where you can identify what parasites I may have and explain what symptoms that parasite would cause. Why do we get those symptoms? Basically, you can't just say, oh, I think you have schistosoma and ascaris, I don't know, malaria. Yeah, that's not enough. You need to tell me why. That one you can get up to 5 points depending on how well you do on describing and identifying everything. The other bit of extra credit is the IDEA evaluations. So starting next week, we'll look to see what our response rate is, but target is to get 90% plus. All right, and those will be extra credit, some extra credit on this exam that we have coming up and the final exam. All right, have a good weekend.